All right, sorry for the disruption, but we'll get back on track here. And my talk was actually about to go into the mating pathway, so that was very interesting, I found. Yeah, and normally those kind of things don't happen with ciliates. So I'd like to introduce Alejandro Saettone. And he's going to tell us about functional analysis of the Ibd1 protein in Tetrahymena.
Okay, so good morning, everyone. So I want to introduce you to our lab research question, which is how do histones sequester DNA from DNA binding proteins, and how does chromatin remodeling regulate access to these sequences? So as you know, we are going to talk about chromatin, so I'm going to do a little bit of background about chromatin. So chromatin is a physiological template of DNA. So as we follow this, the DNA strand here, so this is a cartoon of the nucleus, and within the nucleus you can see the nucleus, chromatin. So this is the DNA, and then this little green piece here represents the core histones, which are H2A, H2B, H3, and H4. So then the DNA wraps these core histones to form the nucleosomes, and then a higher order of packaging. So it's important noting that there are histone variants, an example of which is the H2A set, which is a histone variant of H2A. But what is chromatin remodeling? Chromatin remodeling regulates accessibility of DNA within the nucleosome. So there are mechanisms of chromatin remodeling to ensure that processes such as DNA replication, DNA repair, and transcription occur in the way that they should be occurring. So there are two mechanisms of chromatin remodeling. The first one involves post-translational modifications, and we are going to call them PTMs. And these post-translational modifications, some of them can be methylations, phosphorylations, and acetylations. So I will use this for 2 slides, this little cartoon, so I will try to explain it. So this circle here represents the histone octamer. The orange strand represents the DNA strand. This black box here represents the transcription factor binding site, and this little tail represents an N-terminal tail. So I like to point out this enzyme, which is histone acetyltransferase, an example of which is the GCN5. We can see here, this is a representation of how this GCN5 is adding an acetyl group to the analyzing of an N-terminal tail. The second mechanism involves ATP-dependent chromatin remodeling complexes. These are they are large, multi-protein complexes, and the highlight of this is that they have an ATP-dependent subunit, and they are conserved in eukaryotes. So they can be divided into subgroups. The first one, which is of interest of my work, are histone ejectors or sliders, an example of which is the SWI/SNF complex. And you can see here the transcription factor binding site is being exposed after the core histone was a slide. You can imagine also how this is important for transcription. So the second kind of chromatin remodeling complexes can be grouped in histone variant changers, an example of which is the SWR complex. And here you can see how H2A is being replaced by an H2A set. So these mechanisms of chromatin remodeling are sometimes interrelated, and some PTMs are recognized by the catalytic subunit or distinct subunit of ATP-dependent complexes. Here we can see the acetyl group here, which is the post-translational modification. And the ATP subunit of most eukaryotic SWI/SNF complexes contain a bromo domain. We're going to call it BD. The bromo domain is a distinct protein domain that binds to acetylated lysines. So yeah, this is the same idea. So we slide the histone octamer to expose the transcription factor binding site. So let's talk about now my work. That was all for background for the moment. And which is, we started with identification of the functional characterization of the SWI/SNF complex. So the starting point to identification of the SWI/SNF in Tetrahymena started in 2006. And this work did preliminary functional analysis of ATPase domains, and it found a BRG in Tetrahymena. So one of the important things that was found here, if you remember I mentioned that this ATPase domains normally in Drosophila, human and yeast will have a bromodomain. In this case, they didn't have, this BRG didn't have a bromodomain. And also it was the generation of an antibody against Tetrahymena BRG1. And comparative genomic analysis predicted the existence of other 4 additional members, all of which are part of the SWI/SNF in yeast and humans. So on the left we have the yeast protein, and on the right we have the Tetrahymena potential homolog. So we have the BRG without a bromodomain, and then we have other 4 members, SWI1, SWI3, SNF5, and SNF12. So we decided to tag the SNF5. Why SNF5 is because it's a well-conserved subunit in yeast and humans, and it was a potential homolog in the Tetrahymena. So this is just like the final results. We did 4 experiments of affinity purification, mass spectrometry, followed by a statistical analysis that we have. It's done by JP Lambert. It's called SAINT Express, and it's going to be explained later. Not by me, but by Dr. Perlman, I think. So, well, these are the SNF5 interactions. So all the proteins that you can see here are proteins that have passed this robust analysis. So you can see the proteins that were expressed by Dr. Finnegan in 2006, SNF5, SWI3, SWI1. We had the BRG without a bromodomain, SNF12. But something that we found here is that there is a bromodomain containing protein and a PhD finger. That's not usual. So we have interest in the Ibd1, and we did the alignment of Ibd1 with GCN5, which is a bromodomain containing protein as well, and 2 bromodomain factors in yeast. And we can see here that it has all the parts of a bromodomain, alpha set, A, B, and C, and also has a potential or putative protein-protein interaction domain. So then we decided to tag Ibd1, and we did, again, 3 experiments of biological samples of Ibd1 Ibd1, and we found all these other proteins. And we call it interactive bromodomain, and then we call this the SWI/SNF complex in Tetrahymena thermophila. Then we group this putative saga, this putative SWR, which is related to histone-variant exchange, a histone-methyltransferase, and then these other proteins that cannot be grouped in any of these complexes at this point. The thing that we don't know is that in yeast, okay, I based these groups because they're homolog to yeast, but in humans they work together, saga and SWR, so they might be together, we don't know yet. So let me introduce you to the first Ciliate SWI/SNF complex. We have the catalytic unit, and we have the 2 baits that we use, Ibd1 and SNF5. It has SWI3, SWI1, and SNF12, and also PhD finger. And just to mention that we have some Tetrahymena specific and Ciliate specific, but again, these proteins are disabled because they pass a robust statistical analysis called SAINT. So also this was the beginning to establish members of putative complexes in Tetrahymena thermophila. So then we wanted to look at the functional analysis of the IBD protein Tetrahymena thermophila. So we did some Western blots. On the left panel, you can see our wild type. And on the right panel, we can see the Ibd1 Fz set. We have anti-BRG, which is an antibody that is supposed to be seen during vegetative starvation and mating. And we use also a PD-1 antibody that is purely developmental. And in the string, we can see that Ibd1 localizes throughout the Tetrahymena thermophila life cycle. Now we wanted to see the localization. And if we focus on the right here, we can see that localizes to MAC, which is also related to transcription during mating, after 3 hours, and after 24 hours. So now we wanted to see if this Ibd1 was functional or not. So we used this commercially available peptide array from active motif. They use post-translational modifications of humans. They are single and multiple different post-translational modifications. So we synthesize an Ibd1 synthetic to be expressed in BL21, E. coli BL21, And our top hits were histone H3, acetylation of lysine 9 acetylation and 14, so in conjunction, and also the combination of H4, lysine 5, 8, 12, and 16, which are also related to transcription. So we came out with this model. It's a transcription model in Tetrahymena, which it says that it is generally accepted that highly transcribed chromatin regions in Tetrahymena have multiple acetylations in H3 and H4. Our data suggests a model where promoter-specific H3 and H4 acetylations are recognized by Ibd1, bromodomain to recruit SWA/SNF, SWR saga, and histomethyltransferase, all of which work together to allow access to the RNA polymerase II. And we predict that these Ibd1 containing complexes are responsible for high rates of transcription on a subset of genes during beta-AT growth. So now what we are doing is we are testing the model. So these are all experiments in progress. So we are developing this technique called ChIP for Tetrahymena. We are trying to apply it into our samples. So we have the ChIP qPCR shows that Ibd1 is enriched in the highly transcribed alpha-tubulin. And after this, we went like kind of backwards because we have already the primers. And then we just received this data, the ChIP-seq. And also the first gene that I analyzed was alpha-tubulin, of course. And it shows that there is an enrichment in the promoter, actually, of alpha-tubulin. So the summary, through affinity purification, mass spectrometry of Tetrahymena, SWA/SNF, and SNF interact in IBD, we have identified the first protease SWA/SNF complex. Affinity purification of Ibd1 suggests that it is a subunit of other chromatin remodeling complexes and a histomethyltransferase. And localization of Ibd1 and a peptide array confirms that they are likely related to transcription. So Ibd1 is related to transcription. And preliminary data using ChIP-seq suggests that Ibd1 localizes to highly transcribed genes, and it's consistent with the ChIP qPCR data. So ongoing experiments are what is the role, we wanted to answer what is the role of Ibd1 in Tetrahymena, and what is the composition of the distinct Ibd1 containing proteins complexes in Tetrahymena. And we also want to analyze other proteins that are being copyrifying with the SNF and Ibd1, an example of which is the PhD domain. Thank you for your time.
I have a quick one, actually. Have you tried the loss function study of the IDB1? You know, knockdown, knockout, you know, what's the phenotype for that?
Oh, yes, we haven't, but yeah, we wanted to definitely do the knockout for Ibd1.
If you look through your subunits that are part of these complexes and interactions, do you see some examples of things that are more developmentally specific towards sexual reproduction, or they look like they're mostly all associated with vegetative transcription?
Yes, most of them are, I only did the experiment with SNF5, and we know the BRG and Ibd1, they localize to the MAC only, so they don't localize to MIC. But when we see the experiments, what is in the webpage of Ciliate.org is that they should be overexpressed during conjugation, but we don't see that in the immunofluorescence.
There's also been reports about other acyl modifications or succinyl proteinylation, for example, interacting with bromodomains. Is that was that covered on your array at all?
No.
Did you just focus on acetylation?
No, I mean that array has different post-translation modifications, in total 384, but I'm not sure if it has that specific post-translation modification, but if it didn't have a hit, all the ones were related to transcription.